Good afternoon, everyone, and thank you for hanging around for my talk, I guess. And thank you, Raj, for the invite, and BIOMIN for sponsoring this wonderful symposium. And as opposed to Todd, who only missed one of these meetings, this is my first and only meeting I've been to. So I have lots to catch up on. With that introduction that Raj gave you, I'll preface my talk by saying I got introduced to probiotics as a graduate student at Maryland, and kind of continued to work on that a little bit as a postdoc, but then focused more on it when I moved to Virginia Tech. And that's what I'm going to focus on. I'm going to tell you about a couple of studies that we ran with coccidiosis and necrotic enteritis that our lab and others have run. And quickly to introduce my lab, again, this is the even immunology laboratory, so we focus on a lot of things, mostly poultry health. And we work on a number of potential alternatives, as many of the previous speakers have discussed. And we have a number of applications that we use, and a number of disease models that we have employed over the past few years. So why study all these enteric pathogens and immunity? And that's because it's, as many of the previous speakers have mentioned this morning and this afternoon, not only is it critical to the performance of the bird in terms of nutrient digestion and absorption, but we also have over 95% of the pathogens gaining access to the bird through the mucosal surfaces, most importantly digestive system. So just to give you a little overview of what the kind of defenses that the bird uses, and some are non-immunological and some are immunological. And this is a list that, again, most everybody, and if not everybody in this audience, is familiar with this. And previous speakers have done a nice job outlining some of these factors that play into the defense mechanisms at the digestive level, and including, we all know, the mucins and lymphokines and different cytokines that are naturally produced and continuously produced oftentimes whenever there's an insult in the gut. And also birds tend to have a higher body temperature, so oftentimes they control the growth of some pathogens based on heat sensitivity. pH, obviously, and especially in the upper gut, but also the gut-associated lymphoid tissues, and that's what our interest is in that system, which is actually the largest lymphoid component of the body, whether it's humans or birds, and it houses most of the immune cells. But also, we've heard, as you know, over the past few years, the microbiota and commensal populations, how important they are for the defense system of birds as well as other vertebrates. So I don't need to go again over this. We all know that there is an impetus, so to speak, to look for alternatives, things that could work with birds that maintain performance, yet provide some resistance to pathogens and protective functions. And this is just a short list of things that I thought are important, including the most recent vet feed directive that basically regulates feeding antibiotics or antimicrobials to poultry. So the question is, are potential alternatives available? And as we've seen from literature over so many years, especially the last few, there's quite a few of them that have potential. And again, I have probiotics on red. And red, that's what I've done most of my work. And we like to collectively call them as immunomodulators. And by definition, immunomodulation is a change in the body's immune system caused by agents that activate or suppress its function. So among others, we have the prebiotics, bacteriophages, essential oils, azophytogenics, and the combination thereof. And as many of the speakers have mentioned, it's not just a probiotic and a single species. Oftentimes, companies are looking at a combination of different products that kind of complement each other in terms of optimal performance and immune function. As Ramesh nicely defined, probiotics is a live microbial feed supplement which beneficially affects the host animal, improving intestinal balance. And in many animal species, it has shown to do that, especially under challenge conditions. Now, oftentimes, when you have no challenge and you test a probiotic and you see little differences and you think, oh, well, it's not going to work. And the reason behind that is oftentimes it manifests its benefits when you have an actual challenge, especially an enteric challenge. And that's what we've seen many times over. And the idea behind that is, I believe these slides I stole from my former student. And you're going to use them in your next talk. Okay, thanks, Chas. And it's nicely laid out here when you have like a balance, right? It's critical to have a balance. So you have a physiological microbiota, what we call the commensal microorganisms, or the friendly ones, so to speak. But we also, as indicated, if we went to Lisa Bilke's talk this morning, she mentioned about these constant gram-negative and pathogens that are present in the gut, yet they don't really affect the bird as much. It's only when this balance gets disrupted when you get what we like to call dysbiosis. So you have overwhelming majority of these pathogens disrupting the gut and disrupting the integrity of the epithelial lining and penetrating the different systems through the blood or the lymph. And the idea behind it is when you introduce probiotics or beneficial bacteria, you kind of restore that balance and bring it back to the right balance it should be in any healthy animal. So Ramesh pointed out some of the modes of actions that are numerous, and as he said, you could spend hours talking about and describing different mechanisms, but some of them, and the most important one that we started with back in the 70s is competitive exclusion. And the idea is not only they compete for binding sites on the epithelial lining or mucosal layer as well, and also they compete for certain nutrients. And they naturally produce a number of antagonistic components to bacteria that only impact or kill pathogens, yet doesn't impact the beneficial flora. And we all know with lactic acid and other acids, like volatile fatty acids, organic acids, they naturally favor the environment that these helpful microbiota organisms will survive, yet pathogens will not. And to me, the most important thing is the stimulation of the immune system, and that includes a number of mechanisms, including humoral immunity by enhancing secretory IgA, the immunoglobulins that are the most common immunoglobulins found in mucosal surfaces, as well as priming immune cells, whether it's innate or adaptive immune cells. So there's a long list. I only mentioned a few here, and that about projects and trials and products that were tested in poultry, whether it's turkeys, chickens, and as well as ducks, that clearly show the probiotic effect, enhancing performance and promoting gut health with number of parameters that you could measure. And most of our work has focused on disease challenges and how they modulate the immune system. Yet it's still limited to, as Todd mentioned earlier, to the agents that we have available on hand. So we could do as much as we can with the tools on hand, but we cannot do everything that others on the human side, mammalian side can do, or even on the pig side. And here's a list of some of the pathogens that are most commonly used in these models and listed in these publications that actually were helped by addition of probiotics. Now, this audience knows very well the different genera and species that are typically included. And again, as many have mentioned before, it's not a single genus or not a single species within a genus that makes a good product. It's oftentimes a mix of these products that provide a good balance of colonizing the upper gut, the middle, and the lower gut to provide the optimum benefits to its use. So with that said, I'm going to go briefly over one study that one of my students did a couple of years ago. And we actually worked with Poultry Star on this one. And these are this is the composition of the actual product that we use. So in this one, we decided to look at a coccidiosis challenge, but using two different methods of delivery, water delivery as well as a feed delivery. And as far as the experimental methods, we had COP 500 birds, and we had 6 treatments, and we had negative control, positive control, and an AGP control, and as well as 3 combinations. We had a water treatment that's continuous, water treatment that's intermittent, and as well as a feed addition, supplementation. The challenge model that we used in this case was on day 15. That's when we introduce to individual birds this mixed inoculum of Eimeria, the 3 major species that we typically encounter in the field. And as you can tell, if you're familiar with coccidiosis, these are not the highest doses. So this is just high enough to impact performance, which typically what happens in the field, but it's not high enough to induce very severe lesions and high mortality. So again, these are the experimental groups that we had. So this is the negative, and there's no administration, either water or feed, and no Eimeria challenge. And the positive, only the challenge. In this case, we use Salinomycin as our positive control in terms of having AGP, and we use Sacox at the recommended rate. And then we have a water probiotic supplementation that's continuous, and that was 2 milligrams per bird per day. And as you can tell, Miranda spent a lot of time mixing these products and providing the birds. And then we have intermittent water application as well as a feed supplementation. So quickly looking at, in the interest of time here, we're looking at day 15 to day 21. So that's from the week immediately, or 6 days immediately following the challenge. So typically we raise them on the products for 2 weeks, introduce a challenge at day 15, 6 days later. That's when we expect lesions to be at their highest, and that's when we necropsy birds and measure performance and look at lesion scores and what have you. So if you look at this, we didn't have much differences in terms of body weight gain with the probiotic addition. Did I skip one? No, sorry. But when we look at feed consumption, particularly with the continuous water supplementation, these birds actually ate less, consumed less in general than this Salinomycin. But it wasn't much different. It was not significantly different from the positive control. Yet when we look at FCR, this again, when you look at statistics, it's not that significant, but there are some couple of percentage points reduction in FCRs, particularly with the continuous water supplementation. Now, when we looked at lesion scores, and everybody familiar with lesion scores, the typical, the Johnson and Reed is basically the reference for lesion scoring in terms of coccidiosis. And you go from 0 to 4, and we rarely ever see 4, because by the time you see 4, lesion score of 4, the bird is dead. But you're looking at the Wallen lesion. This is a score of 1, a little bit of necrosis here, and this is obviously duodenum, so it's Eimeria sylvanina. Score of 3, you can see the sloughing of the epithelial layer. And looking at our treatments here, again, I keep pointing out the continuous water supplementation, and that's what correlates well with the performance, where we had the lowest lesion score. And that was, again, this is not the most severe infection model, so we're looking at 1.5 or anywhere between 1.2 and 1.6 or so in terms of average lesion scores in the positive controls. And acetaminolysin obviously reduced that, but this was even better by the supplementation of in the water, continuous supplementation in the water. Also looking at the jejunum, which is a site of infection of Ehrlichia maxima, same trend, but this time with both water treatments reduced lesions significantly in comparison to our positive control. We didn't see any lesions in the ceca, and that's Etenella, and if you remember, the Etenella dose was actually very low, and it varies sometimes when you have a more pathogenic strain than other, and in this case, we didn't see any significant lesions throughout. We also looked at histology, and we did microbial profiling. I don't have all the data on that, but we also looked at gene expression of an array or a panel of genes that are related to the immune response to coccidiosis. And these are roughly, we try to categorize them as innate or Th1 and Th2. Yes, these are Th2, but oftentimes they actually cross between the different categories. Of note here, looking at interferon gamma and INOS, and these INOS is basically inducible nitric oxide synthase. What it does, it induces the production of nitric oxide, which actually kills the parasites. And interferon gamma is one of the premier cytokines that respond to coccidiosis and controls it. And incidentally, when we're looking at day 21 in the jejunum and ileum, we had much higher expression of those two genes, twice as high than the positive control and the anticoccidial control. But also at the same time, some of the pro-inflammatory cytokines were reduced on day 21 in the jejunum and the cecal tonsils. So what we concluded out of that is not only enhances the probiotic, enhanced the innate immune response during the infection when looking at INOS, but it seemed to have shortened the inflammatory response by reducing IL-1 beta and interleukin 12 B, and it also downregulates, by downregulating the gene expression of these genes, it helps reduce the severity of the challenge. So overall, the probiotic supplementation without using an anticoccidial, this is all in comparison to that, modulation intestinal gene expression, and we basically believe that's partly why it alleviated the lesion scores. So in summary for this trial, the probiotics via water basically outperformed probiotics in the feed in this study, and also again without the anticoccidial we were able to see some, albeit not significantly different from the anticoccidial, some enhancement in performance, and it clearly alleviated the negative effects of coccidiosis. So, and the next question that we wanted to test is, how does it work with coccidiosis vaccines? And they may not be the most popular or the most commonly used in broilers, but they are used in breeders as well as layers. But many operations based on what area you're in, they use coccidiosis vaccine. So what that does, it gets administered the first few days. It's typically either at the hatchery or right before placement through spray cabinet, or you provide it in the water as you place your birds. And it provides a neatly uniform administration, and these are the different methods that are typically used in the field. And what this does, you're providing a very low dose of live oocysts. Very low dose. You're talking like a few hundreds. And what it does, the birds will pick it up, and they will cycle the oocysts through their system, and then they shed it back in the feces, and they pick it up again from the litter, and the cycle goes on until a couple of weeks when immunity actually develops. So what that does is you're providing immunity, but you're impacting early performance of the birds, and that's been the negative connotation with this application. So our question was, can the probiotic, if applied early on, help the birds deal with this early lack of performance or reduced performance, I shouldn't say lack of performance, as a result of vaccination? So we designed this a simple study using, again, one of the commercial products here with this composition. And in this case, we used Immucox vaccine in collaboration with SIVA that makes the vaccine, and it contains the cervellina. That's the duodenum, maxima, jejunum, Necrotic, that's the ileum, and tenella, it's the ceca. And what we did, that was, the vaccine was given at the hatchery and through a gel droplet application, and then we used the same challenge model, but this time we used the 10x the dose of the vaccine, the regular dose of vaccine, as our challenge. So it was a grow-out trial with many less birds, 400 birds. We had 6 reps in this case. So we had a challenge control, a vaccine only, probiotic only, and then we combined it. So it's simple for treatments. They're all challenged. And again, here's the 10x dose of the vaccine at day 15. And here's a summary of some of the data we observed. So when you look at day 15 before, this is 2 weeks or 15 days after the application, whenever you had the probiotic, whether it's with the vaccine here, without the vaccine, you had a better body weight, higher body weight than the vaccine alone. It's not different from the control. At day 21, we see the same trend, especially with the vaccine, without the vaccine, the probiotic only. But at day 28, by the time the birds have recovered from the challenge, or mostly recovered from the challenge, you see clear body weight differences among the treatments. And then in looking at gain, you see the same trend, and I have this highlighted here or circled, and when you add the vaccine and the probiotic, put them together, and the group, you see the differences in terms of body gain compared to the vaccine only. Same thing from day 21 to day 28. And that actually is reflected in terms of the FCR. Again, the probiotic treatments had much better FCR than the vaccine alone, whether it's without the vaccine or with the vaccine. And that's early on before the challenge, and this is the first 2 weeks, and that's what we were talking about in terms of the challenge induced by the vaccine at day of hatch. And then again, you could oops, sorry. Imagine the trend also going with the lesion scores, and so that's the control here, the vaccine only, the probiotic only, and this is the most interesting component, which is the vaccine with the probiotic. This is a cervellina, and we barely see any lesions there. Reduced lesions, but only in comparison to the control, not much different from the other 2 treatments. And again, with the ileum here, which is Necrotic, we didn't see much differences at all. So in summary, the early probiotic application, especially the water application in this model, because that's what we used, was effective in alleviating the impact of the Coccidia infection with the vaccine challenge. And we concluded that when you combine the two, actually provides an enhanced protective effect against Coccidia challenge with the water application of probiotics. And so we think it may enhances these defenses because it helps maintain the integrity of the epithelial layer that we've talked about earlier, and thus improving the digestion and absorption of nutrients, and that's what kind of limits the early growth suppression that you see only with vaccines. So I'm going to switch gears here and talk about a couple of trials with necrotic enteritis model. And this was done back in 2009, and it used a specific floor model with a vaccine. And this was done later on, which is most recently last week at Southern Poultry Research Lab. So going quickly through the, how am I doing on time? Are we good? 5 minutes? So, okay. So in this case, we had Ross broilers, and they were only going to day 25. And then the unique challenge model here was they use a vaccine, IBD vaccine, infectious bursal disease vaccine at day 14, and then they orally inoculate Clostridium perfringens at days 17, 18, and 19. In this case, they split the study in two major groups. One was giving a low dose, so to speak, and one was a higher dose of Clostridium perfringens. And they had a total of 100 birds per treatment. They get negative control, no challenge, no additive positive control, just a challenge. And the probiotic was given in the water at a recommended rate. And they did necropsy and bacterial counts on day 25, about a week or so after the Clostridium challenge. And most of you are familiar with lesion scoring, kind of like same scale as Cocci, but they're clearly different scores. Different lesion types that you see with necrotic, from the simple necrosis to terry towel type lesions with lesion 4, and that's typically, again, you see them when the birds are dead. So, keep clicking the wrong one here. So looking at the results, lesion scores, this is the positive control, and then you add the soluble type probiotic, and you see a significant drop in lesions. In terms of mortality, 26% versus 6%, and this is the low dose, the 10 to the 5 Clostridium, and also the recovery of Clostridium perfringens also much reduced with the probiotic. Now we're still seeing similar effects on the higher dose, but it was slightly less effective. Yet it was still significantly different in terms of lesion scores, from the positive control to the probiotic. So in summary, there was a clear reduction in the severity of Clostridium perfringens here in this specific model, and that was effective in both of the low and high dose experiments that were run. And the subsequent study that was recently done at Southern Poultry Research, so this was a battery trial, and the probiotic actually was supplemented in the feed. Again, very simple design. You have a negative control, no challenge, positive control, the challenge, and the probiotic was given at 500 gram per ton. And then this is the model that Greg Mathers employs. He gives a 5,000 Iberian maximum oocysts at day 14, and he comes back a couple of days later and gives an oral challenge of Clostridium perfringens on 3 consecutive days. And they looked at performance, day of hatch, 21 and 28, that's what the end of the trial, mortality and lesion scores. And when you look at weight gain, so this is day of hatch, day 21, and this is total day of hatch, day 28. So you don't see a whole lot, but it's still statistically significant in terms of weight gain being reduced in the positive control, which is expected, but that was offset by the addition of the probiotic. And when you look at feed conversion, that's a clear significant differences day 21 and day 28. And then you look at lesion scores and mortality, again, significantly reduced. Mortality didn't have much in terms of statistical significance here, but because you look, it was only 3% mortality. The intention was not to induce high mortality, just enough to induce lesions and disease. So overall summary of all these experiment is, whether it's Eimeria or Clostridium, there is clear indication of improved performance, weight gain, and performance to varying levels based on the model being employed. You reduce mortality and lesion scores, and both the water and the feed, although in these studies presented here, the water was evidently more effective in improving the conditions when you have coccidiosis or necrotic enteritis challenges. So some of the implications and what we be looking at from an immunology perspective, we need to better understand the mechanism of action. Again, like Ramesh said and Lisa early on, there's so many factors involved. It's a multifactorial problem, whether it's coccidiosis or especially when it's necrotic enteritis. There are a lot of things that we need to be looking at. And you got to identify the most efficient strains as well as the formulations, putting the strains together to actually have optimum performance and optimal benefits to the bird, as well as the application methods, as you have seen in terms of water versus feed, but also timing is critical. And Lisa Bilke earlier this morning, she mentioned something about that, and she showed that she can establish some of that in terms when you provide a single species or of probiotic in ovo. And actually it was Chastity, for her dissertation project when she was in my lab, who did the conceptual type proof that we can provide probiotics in ovo and it doesn't impact hatchability, and also you can have a lasting effect post-challenge, although she didn't go throughout 42 days, only 21 days, and you have a lessened or reduced effect of coccidiosis challenge. Again, so it's so complex, and I show this, leave you with this slide just to show you in terms of immunology side only, it's this complex. You're looking at different cells, different immune cells in the lamina propria, different effects of all these probiotics and pathogens. Now you factor in all the tight junctions and everything that Todd talked about, and some of the Ramesh mentioned, and Lisa early this morning. It's very complex. So it's a challenging field to study, and hopefully we continue to generate more tools to be able to study it better and understand it better. With that, I'd like to acknowledge my lab members, Miranda, who did most of the work, and my collaborators, and also BIOMIN for supporting this work, as well as SIVA for providing the vaccine. And with that, I'll entertain any questions if you have them. Thank you.